Hello everyone, I'm Kavita from first year MSc Botany. Here I'm going to present my e-content regarding the concepts of paleobotany. Paleobotany is the study of the preserved vestiges of plant life of the geological past. The remains of past plants are called as fossils. The most convincing evidence of plant evolution is the fossil plant record, which is helpful in the study of past plant life. Fossil plants are usually found in stratified rocks, which are built up of sediments that accumulated in the bottom of lakes, lagoons, seas, swamps, subsiding beaches, and flooded valleys. These stratified rocks got exposed by the natural erosion and released the plant fossils. Man has also been obtaining them by means of drill holes, mine shafts, quarries, and railroad and highway excavations. The word fossil came from the Latin verb fodere, which means to dig. It is essential for fossilization that the whole organisms be buried alive or soon after death without decay by bacterial, fungi, and termites. Only a small number of plants become fossils, but most of the plants either decay and are eaten away by the animals. The suitable condition for the fossil formation occurs such as in bogs with very cold or acidic water and low oxygen supply, which stops the decay of plants. There are two theories of preservation. One is molecule by molecule replacement theory. According to this theory, the original substance of the plant body is supposed to be replaced molecule by molecule by the mineral in solution. There are some instances where the complete replacement has taken place. These replacements are due to complete hydrolysis or weathering of some type of original organic substance except the cell wall material, particularly lignin and cellulose. The replacement is actually gradual and is supposed to be complete when lignin or cellulose remains. The organic substances gradually diminish due to chemical breakdown and molecule by molecule replacement by some mineral substance like iron pyrites, lime carbonate, or silica. Inorganic matters, with exception to water, are perhaps unable to react chemically with wood constituents. The next one is the infiltration theory. The theory is based on the fact that the mineral matters are precipitated and infiltrated in the cell walls. The buried plant or plant part undergoes partial disintegration and results in the release of free carbon, which produces the sulfides present in the water and leads to the formation of carbonates of calcium, magnesium, and iron. Finally, the silica and other compounds are deposited within the cell walls through infiltration and precipitation. The theory can be emphasized by the peel method of preparing thin section. The process entirely dependent upon the presence of organic material within the cell walls. The plants are preserved in earth crust in various ways. Various kinds of preservations require their own technique for study. According to the nature of fossilization, the fossils can be classified into some of the following types, like compression, impression, incrustation, petrifaction, compaction, and amber. The first one is compression. This is the most common kind of preservation in which much of the organic matter of the plant is preserved with the impression of the plant. Here, the organic matter retained on the surface may be a layer of structureless carbon, although sometimes the cell pattern of the cutinized epidermis is retained. Compressions are formed as a result of great vertical pressure of the sediments. The buried plants or a plant part are highly compressed due to the weight of sediments lying over them. Coal is usually considered to be the complex kind of compression formed by an extensive accumulation of the plant material that is gradually compressed in a basin of water. The study of coal under reflected light by grinding thin section or by maceration reveals the presence of cuticles, wood fragments, bark fragments, marking of leaf, and resins. These include most of the familiar carbonizations as evident from the fact that the large percentage of coal was laid around 250 million years ago, the Carboniferous period. Compressions may be studied by film transfer or maceration. One of the examples for compression is Glossopteris accusin godbonastrata. The next one is impression. These are the prints made by plants or plant fragments which came in contact with the soft clay. When the sediments increase in thickness with entombed plant material, compression results and the less resistant and more compressible plant part is flattened to a mere fraction of its original thickness. A leaf falling on semi-stiff clay after its original matter has decayed leaves the imprints of its form and veins. The plant material disorganizes and leaves the impression when the clay gets converted into the rock, gives a superficial resemblance of the fragment preserved. These are often darker in color than the surface of rock below because very often they retain some of the organic material. Some of the specimens look extremely beautiful like paintings or bas relief of the actual twigs. The structure like stomata are clearly seen in very good preparation of well-preserved specimens because of intact epidermis. No cellular details are left in this impression. Here is an example of fossil impression of the cycad leaf found in the California. The next preservation is through incrustation. These are one of the commonest type of fossils. In their formation, the plant part gets covered by sand or mud. Then the covering sediment of the mold becomes hard and the plant material inside rots away, leaving a hollow. The space formerly occupied by the plant, which now is rearrested by a hollow, gets filled up by some rock-forming material. Furthermore, the inside as well as outside solidifies into the stone from which the external part may be peeled off, leaving an exact cast of plant part showing all its surface features. These actually do not contain any of its original organic material of the plant or cellular detail. The surrounding material of the mold forms the incrustation, while the rock material surrounded by incrustation is known as a cast. A cast are valuable to the paleobotanists in reconstructing the external appearance of many kinds of plant. Casts of whole trunks, stump or seeds are usually formed when the plant part decays. The next one is petrifaction. That is also called as mineralized plants. These are interesting types of preservation that allows the study of internal features of organism. These preserve their external form, internal structure and sometimes substances of the original plant. They are formed by involving a molecule by molecule replacement of the plant part by soluble minerals that eventually harden. These are rare and are only means of transformation of the organic tissue into the stones. Although the actual process of petrifaction is not very well understood, it is thought that the buried plant material absorbs mineral solutions like carbonate, sulfate, silicate, phosphate, etc. The infiltration is followed by precipitation so that calcium carbonate, magnesium carbonate, iron sulfide, silica, etc. get impregnated within the plant tissue. Silicified woods, bits of stem, twigs, seeds, and sporangia and coal balls are some common examples of petrifaction. Coal balls are irregularly or subspherical masses of mineral matter, usually of magnesium or calcium carbonates, which vary in their diameter as well as in their weight. Sometimes delicate parts remain intact in the coal balls so that the internal morphology as well as external morphology is clear in this type of petrifaction. Next one is compaction. Compaction is also called mummified plants. These are the plants or plant fragments compressed by vertical pressure, masses of plant fragments without intervening matrix such as occurring in peat, lignite and coal or large-scale compactions. Occasionally in leathery leaves or tough fruits, the tissue are retained in a mummified condition. These can be embedded and sectioned for the microscopic studies. In the amber preservation, the resinous excretions of certain fossil coniferous trees which float due to injuries caused by boring insects or decaying branches. These often drop to the floor of forest, gradually harden and accumulated over long ages. Besides the insects and small animals, these enclose plant fragments, some of which are well preserved and are ranked as fossils. In fact, any object connected with ancient organisms is considered as fossil. Diatomaceous earth formed by the skeletons of billions of diatoms depositing on the seabed, graphite used in lead pencils, different type of algal limestones and even petroleum are considered as fossil. Sometimes certain minerals during their formation or crystallization resemble some plants or animals. There are instances of these being considered as fossils, but in strict sense they are termed as pseudofossils. Here there are some of the techniques for studying the fossil. First one is thin ground section. This technique is employed in case of petrifaction like fossilized wood and coal balls. Coal can also be studied by grinding thin section. Thin sections are prepared from the petrified plant material and then grinded till they attain the requisite thickness and become translucent, thus making the cellular framework of the tissues clear. Next one is film or peel technique. The film or peel technique is the method by which serial sections can be taken out from the petrified specimen. It is of utmost importance in case of small fragmentary material and those which need serial studies. During the process, first the surface of material is etched with a mineral acid, then a commercial acetate film or solution of nitrocellulose is spread over the etched surface and now it is peeled off and it is dried. The film transfer technique has also been used with excellent results on compression, particularly mineral with calcite, bituminous and anthracite coals and fine grained sediments. The film technique is more advantageous than the ground section because the former is more translucent, thinner, less expensive and quicker to prepare. The only drawback in film technique is the trapping of small air bubble which often interfere during high magnification under microscopes. The maceration technique. It is commonly used in cases of peat, lignite and coal. During the process, the specimens are treated with the mineral acid or other suitable reagent which separates large fragments is made free from associated rock and cemented to a glass slide. The exposed surface of a glass is coated with paraffin wax and then it is treated with some mineral acid to remove mineral matter. The specimen, after being washed, dehydrated and mounted, is ready for the study. The process, although slow, causes least destruction to the fossil, but the specimen is usually dense and opaque. Microtome method. The specimen is kept for few weeks in hydrofluoric acid and potassium chloride and then treated with a mixture of equal parts of phenol and 95% alcohol. Finally, it is embedded and sectioned with microtome. Because of prolonged process of demineralizing and embedding, the method has not received proper attention. Excavation technique. The specimens of plant fragments are removed with microvibratory machines or with a small hammer and steel needle. The process being slow and tedious, sometimes giving excellent results for the minute leaves and sporangiophores. Calmophyton was obtained by the excavation technique. Electron microscopy. The observations made by Wesley and Cooper on the xylem element of lepidodendron were based on electron microscopy. The specimen obtained by this technique showed the presence of cellulose and lignin in the cell wall, but is commonly not employed due to complex equipment and intricate procedure involved. X-ray radiography. The plant fossil, when photographed by X-rays, exhibit differential shadows corresponding the density. A shadow picture taken by the sensitized film, that is radiograph, can be used for fossil study. The technique was used by Dara during his study of certain calcified seeds from the coal balls. Chemical method. The basic principle underlying this technique is to identify the constituents of fossil plants. The method has proved valuable in tracing the presence of chlorophyll and hemoglobin in carbonaceous sediments and carotenoids in the lacustrine muds from the post-Pleistocene lake near Moscow. C14 method is most valuable technique for the study of fossils. Here there are some preservation methods. Carbonization is a process where only the residual carbon of the organism remains. In the nature, this usually happens over time when the organism is subject to heat and pressure. A very common example of carbonization are fossil plants, where only a thin carbon layer is left on the piece of shell. In the Carboniferous time period, vast fern forests created miles of carbon, which we mine today as coal. Another most recent example is the fossilized feathers found on dinosaurs in China. These are left as carbon imprints in the shell around the mineralized dinosaur bones. Replacement is similar in that minerals seep into the organism. However, the mineral replaces the original organic material as the organic material rots away. In the end, the organism is replaced by the minerals. Recrystallization involves a change in the crystal structure but not in the mineral chemistry, similar to recrystallization in metamorphic rocks. For example, the mineral aragonite, a common mineral of many shells, sometimes changes to calcite, a more geologically stable form of same chemical composition. Calcium carbonate. Typically, the overall size and shape of a recrystallized fossil do not vary substantially from the original unaltered specimen, but fine details may be lost. Permineralization. It occurs in a porous tissue such as bone and wood. In this type of preservation, minerals dissolved in the water such as quartz, calcite or pyrite permeate the pore space and crystallize. The addition of these minerals results in the denser and more durable fossils. The original bone or wood material may be preserved, or it may be replaced or recrystallized. Unaltered remains. An organism is considered unaltered if there is no change in the original composition of the organism. An example, it includes insects and small animals trapped in amber. The sap from once living tree entombed the animal. This sap eventually hardened and the original animals are preserved inside the amber. Next, paleopalynology. It is a branch of palynology that is the study of science of pollen and spores of plants, which include the study of fossil pollen and spores. The fossil pollen are found abundantly in the peats and also in the alluvial soil and even in strata below. They occur in stratified layers, making it possible to calculate their age. Their study is quite useful in tracing the history of plant communities, showing the climate at the time they were shed. It also facilitates the study of possibilities of exploration of oil, coal, and other minerals. Next, determining the age of fossil. Fossil formed in the past are embedded in the earth layer of different ages in a geologically undisturbed sections of the earth crust, the deeper layer or the oldest layer. High-lying land erodes material which gradually piles up on the lowland and on the sea bottom. Fossils embedded in successive layers thus provide a time picture for evolution. The actual age of rock layer is determined and excellent clocks are built right into the Earth's crust based on the radioactive substances. The rate of disintegration and the end product of disintegration of these substances are accurately known. A given quantity of radium is known to decay into lead in a certain span of time. When these are found together in one mass within a rock, the whole mass presumably had been radium originally when the rock was formed. A similar principle underlies age determination by potassium organ dating and by radiocarbon dating. In the former, one measures how much of the unstable isotope potassium has decayed into the isotope organ 40, whereas measurement of carbon 14 is an isotope natural carbon 12. The potassium organ method can be used for dating fossils many millions of years old, but carbon 14 method is accurate only for fossils formed within 15,000 years. Ages of plant life. Based on the data obtained from the fossil and radioactive clocks, it has been possible for geologists to construct a geological timetable indicating the age of successive Earth layer and provide a calendar of Earth's past history. This calendar consists of 5 successive main divisions of eras. The last 3 eras are subdivided into a number of successive periods. Here there are main events of Earth history. Plant fossils are of importance insofar as they furnish an insight into the history and past geographical distribution of the plant groups, introducing us to many extinct types. It was during early Precambrian, about 4.6 billion years ago, and the Mesozoic era was an age of gymnosperms, as it is evidenced by the abundance of Psychrophyceae and conifers. At the end of Mesozoic, flowering plants underwent a phenomenal development to become a dominant plant group of today. Thank you.